Hello, and today I'm going to speak about the 4 vaccine types and the new corona vaccine. To get an idea of how a vaccine works, it is important to get how the body actually responds to a bacterial infection or a viral infection. And here you can see the innate immune system and the adaptive immune system. And the innate immune system is very important, even though it's not actually able to adapt to specific viruses as a vaccine actually does. But it has a rapid response, and thereby it is able to help your body much faster than the slow response immune system. And here in the rapid response system, we have the macrophages, which are the cells that just kill anything that they come in contact with, which doesn't seem to be right in its place. And the dendritic cells are kind of like the immune system's brain and are telling what to do to the different cells by bringing them like a message, and it goes back and forth between like the innate system and the adaptive system to give the message from what these cells has found. So like the macrophage meets a virus, and then it responds by eating it or destroying it, and thereby it shows on its outside what kind of virus it has been eaten. And therefore the dendritic cell can see, okay, you have been eating this influenza virus, let's say that. And the dendritic cell can tell all of the other cells, both in the rapid response system and in the slow response system. But the difference is that when the dendritic cell tells all the B cells what they have encountered over here, the B cells will start and produce these antibodies, and they will make the T cells adapt by recognizing these antibodies and thereby already knowing what is going on over here without even have met it, and it will be able to attack, and it will be able to adapt into T cells here and these T cells, and it is like a memory T cell or a killer T cell, and thereby they can like adapt. And the B cell does the same thing. It divides into plasma cells or some B memory cells, and the B memory cells remembers the virus, and thereby if you get attacked another time, you will be able to respond much faster and like kill off the virus or the bacterial infection, if it was a bacterial infection we were talking about. But most vaccines are against viral infections, since we can't cure viral infections with antibiotics, which we would usually use with bacterial infections. So now that we know a little about how the immune system works, we are going to talk about the 4 main types of vaccines. And we have this type 1 vaccine, which is the live attenuated vaccine. And that means that they have taken the virus you want to make a person immune to and weakened it. So when you give the vaccine, you give like the same virus but a weakened version, so your body is faster and quicker at killing it off, and thereby making you immune from an infection later on if that should occur. and it is kind of in the same category as the inactive version, because that is also just the same virus as you want to kill off, but this time it's inactive, so it is not a live virus, it's actually dead. And mostly with these vaccines, it's seen that we put other things into the vaccine, such as aluminium, because your response to a dead virus is not really existing, so thereby you um, put another activator into the vaccine, so your body knows that it is not supposed to have aluminium inside, so it tries to kill off um, the vaccine, and thereby it also recognizes this inactive virus, even though it isn't actually dangerous to the body since it's inactive. And these two vaccines are very common to give when you want to fight off flu infections or influenza. And then we have this third kind, which is the toxoid um, version, and that is actually when you give like an actual toxin in small doses to be able to fight off an toxin. And this is often seen with if you get like a snake bite, you get a toxin inside. So this type of vaccine is often used in to fight off like a toxin um, attack on your immune system. So it's not like a virus in this case. And then the last type is the recombinant protein. And that means that it's not really the whole virus that you're putting into the vaccine. So if you can like tell from this picture, you can see this, which is the spike protein. So if up on this picture with the live attenuated vaccine, you can see the whole virus and these small balls, which are actually the spike proteins that are on the outside. And that is the one thing we can see here. And that is the thing that the virus uses to attack your body. So it that is the way it like attaches itself to other cells in to be able to like attack them and take over the cell and reproduce itself. So when you use like only the spike protein, you are actually able to make the virus recognizable for the cells, or they can see it as a threat, and it can start producing antibodies against the virus, which is kind of cool because you don't need the whole virus, you only need like the spike protein. Now to what's going on in the world right now. The SARS-CoV-2 vaccine. It has been developing as the fastest developed vaccine ever in history. And it actually uses two completely new methods. But they do use, both of them uses DNA from the spike protein, as we were speaking about right before. The first type is the mRNA vaccine, or the messenger RNA vaccine. This uses the messenger RNA, which is a single-stranded DNA, which is used to tell a cell what protein to produce. And therefore they have targeted the exact strand of DNA which tells the cell to produce the spike protein. And by taking this DNA and taking the single-stranded messenger RNA, they have put this into the vaccine, and when you inject that into the body, your cells would start taking this messenger RNA, and it was starting to produce the spike protein as we saw before, and your body will respond to the spike protein and start to produce the antibodies to fight off an potential virus infection. There is some downsides to this method though, because messenger RNA is very unstable, and thereby you have to keep it very, very cold, and it is because it breaks down very easily, so it is a very difficult vaccine to produce and handle. This method is what is used in the Cypher and the Moderna vaccine used against SARS-CoV-2. And another method is the adenovirus. Oh, it spells wrong, but it says virus vaccine. And this vaccine uses actually the whole DNA for the spike protein. So not only the messenger RNA, but the whole RNA. But the difficult thing about this method is that it has to use a harmless virus carrier. So it has to be the DNA is put into this harmless virus that then takes over some cells and starts producing these antibodies, and thereby you can get an immune response to the antibodies. But the difficult thing is that this carrier, your body is actually also going to start producing antibodies against the carrier, and thereby you will get immune to the carrier. So the carrier has to be swapped out eventually a lot of times, because you can't keep using the same carrier. So if you have to be vaccinated multiple times, you would have to swap up the carrier, or if you would be to use this method for other vaccines, you would have to use other carriers so that it would work. And this method is what the AstraZeneca and the Johnson & Johnson vaccine is using. And here at last you can see my sources, and I also used my own notes from Gabriel's classes.